The apparently modular architecture of PKS and NURBS megasyntheses immediately makes one wonder what happens when you mix and match the modules or domains. In this review, Kosel and co-workers describe examples of modularity amongst megasyntheses. It is very clear from examination of the sequences of these biosynthetic pathways that the organization of modules and domains is in some way modular. Bioinformatically, it is very common to observe that structurally related PK and NRP enzymes are chimeras of two or more ancestral pathways. It is also self-evident that the junctions for the chimeras are consistently at domain and module boundaries. So what would happen if I swap the position of two modules in the megasynthase? For example, what would the product be if I flip the positions of module 2 and module 5? A modular system may be defined as a multi-component system that can be divided into smaller subsystems, which interact with each other and can be predictably interchanged for functional flexibility and variety. Let's consider an example PKS gene cluster. There are many scales at which you might find modularity: the entire native biosynthetic cluster, the refactored set of proteins associated with the gene cluster, the individual coding sequences, and finally the individual domains. The question is, which of these scales are truly modular? Where are there well-defined interfaces that can be recognized as being orthogonal or equivalent to one another? These biosyntheses are clearly modular around the production of precursors and cofactors. For example, if your PKS needs propional-CoA, you don't need to use the native genes to make that precursor. Any path to that soluble intermediate will satisfy the design. This review is asking about the domains and modules. Are those interchangeable too? Most attempts to make chimeric PKS designs have resulted in low or no yield of the chimeric product, though there are successful examples. It's usually not a folding problem. The modules themselves tend to retain structural and functional integrity as standalone proteins. You can express just a module in the cell, fuse it to others or not, and its expression tends to be contained within the module sequence and thereby insulated from higher order engineering. Similarly, the ketosynthase Ks, acyltransferase AT, and acyl carrier protein Kr and DH domains of PKS modules all retain structural and catalytic integrity as standalone proteins. Thus, based on simple logic that we would use for monofunctional enzymes, PKS systems would appear to be functional at both the domain and modular levels. They express and they maintain their ability to perform chemistry as individual proteins. Where things go awry is in the communication between the modules and domains. Here I show the crystal structure for one of these modules. You can see how the ketoreductase and acyl carrier protein are spatially positioned around the ketosynthase domain in the middle. When you look at these systems at the primary sequence level, it is easy to forget that it is an intricate three-dimensional molecule. The difficulties in mixing and matching these things is usually attributed to mismatches in the linker regions between the domains and between the modules. We'll talk a little bit more about that in a bit. However, it could also be attributed to a lack of true naivete towards the substrate. For example, a module in principle could examine the stereochemistry of a unit added 2 steps prior and reject the substrate. If such specificity exists, it would be very difficult to recombine the modules. It is also possible that the system became canonized. This word, canonization, refers to the situation in which a gene has been isolated within a genome for so long that its sequence is drifted and it no longer operates under the same principles as its siblings. For example, the linker regions might have evolved a new fold that isn't compatible with the linker regions of the siblings. Kosel attributes much of the difficulty in making chimeras to the linkers. These linkers make up a substantial portion of the polypeptide's overall length, and there are extensive contacts between the linkers and catalytic domains. Some of the linkers appear to be universal, but not all. The ones that join module to module appear to be more universal. Thus, it is usually easier to make chimeras at the level of modules than at the level of domains. If you change the domain-domain junctions, usually there is miscommunication between the domains and a rate impairment. Trying several linkers often fixes the problem, resulting in functional chimeras. Another approach is to hold the linkers the same, but just swap out the catalytic portions of a module. For example, you might swap an R enantiomer generating KR domain with an S generating one, but retain the native domain's linkers. Regardless of the initial chimera, it is usually necessary to do directed evolution to fix an otherwise non-functional interface between two domains or modules. Using some fairly simple reasoning, we can write out the domains and modules required to build a particular polyketide. Let's try an example. Suppose I want to make this polyketide. I know PKS builds 2 carbons at a time, and it's always 2 at a time, so moving back from the acid 2 carbons would put the new bond here. There aren't enough carbons here to have 2 steps of PKS synthesis, so this is just going to be the initiating unit on the left and an extending unit on the right of this bond. The extender unit appears to be methyl-malonyl-CoA since there is a methyl group dangling off. Let's focus on the new bond. We see that we have an alcohol and it is not methylated and it didn't get eliminated. This suggests that the module had a Kr domain for the reduction of the ketone to the alcohol, but it lacked a Dh or Mt domain. We can also see that this is the initiating module, and there would be a methyl side chain on the initiating carboxylic acid, so it must have been propional-CoA. All modules have the At, Ks, and Acp domains, so our module would look something like this. The devil is all in the details. In this case, arbitrary choices for these parts usually results in no function, but some combination of parts will result in the desired function. The difficulty is choosing the exact correct combination of domains and linkers to make it all work in concert. This is the essence of the functional composability problem. That is, how do you determine exact combinations of parts that together do exactly what you want them to do? Ideally, you'd want to set a sort of biophysical-based algorithms or heuristic tools that allow you to make these sorts of predictions. One challenge is that we're often talking about issues of 3D structure and the prediction of interactions, and the algorithms might require high-resolution crystal structures to be meaningful. Such modeling is moving quickly, but this today would challenge our abilities. It's not entirely clear, though, that such models are required for this particular problem. The alternative is to do a lot of combinatorial libraries or directed evolution. This strategy does work, and we'll see an example of that next, but it is a much more expensive experiment.